All right. So here, as you see, yes. So here, as you see, I told you meiosis happens in the sex cells, okay, or the germ cells we call it as. So ये main difference है that you need to know. So mitosis occurs in the somatic cells. So what is somatic cell? Give me a second. Okay, so just
Student, I think I'm now audible. Can you tell me whether I'm audible? I'm audible now, right? Okay, okay, awesome. Thank you so much, guys. Sorry for the inconvenience. Okay, okay, fine, guys. In the today's class, we are going to mainly start the cell cycle and the cell division. Okay, you all know how the idea about the prokaryotic cell and the eukaryotic cell, right, guys? The prokaryotic cell is a single cell, does not contain any cell organelles, right? Whereas the eukaryotic cell contain the membrane bound organelles. Okay, can you tell me, guys, which is a common in the both the prokaryotes and the eukaryotes? Which is common in both the eukaryotes and prokaryotes, guys? Which organelle? You can tell me. Okay. So in the starting, what we can do is let us solve some of the cell questions. Okay. Then we'll go to the cell cycle and the cell division chapter. Okay. Because we should have some basic idea about the cell. So that's why I'm talking about the cell now. Let us solve some questions. Then we'll move to the cell cycle and division. Okay. In the today's class, I brought all the questions which are from NEET 2024 and the re-examination 2024 and the 23 exams. Okay, fine. You can answer to this question, guys, which is the correct answer. Very good, Raju. Yes, Rutik. Ribosome is a common. Yes. I will launch the poll. Okay, kindly answer to this question. Guys, here in the match the following, most of the cell chapters, you will get like the match the following only. In one side, they will give the scientist name. In the another side, they will give the organelle name like that. Okay. Or one side organelle name and one side functions. Okay. Fine. I'm going to end the poll. Yeah, sharing the result. 67% given the option A, 17% B, and 17% C. No problem. Yeah. Which is the correct answer, guys? The correct answer is A. Okay. The correct answer is A. And here you can see, guys, Fleming. Okay. Fleming is a scientist who discovered the chromatin structure. Okay. Chromatin. Then coming to the Robert Brown. Robert Brown, who discovered the Robert Brown is the nucleus. Okay. Nucleus. And you can see here, B is the 4. And George Palade. George Palade is the ribosome. Okay, then Camillo Golgi, you know that a disc-like structure, cisternae, is present in Golgi apparatus. So that is discovered by the Golgi. So the correct answer is A. And you can take a screenshot of this table. This is very important. Okay. Robert Hooke identified the dead cell and the Anton van Leeuwenhoek living cell, and the Schilden and Savan given the cell theory, and Rudolf Virchow is the scientist who told that the cell come from the pre-existing cell, means omnicellular, e-cellular. And fluid mosaic model given by the Singer and Nicholson, there is cell membrane, and the nucleus structure is given by Robert Brown, Golgi, Camillo Golgi, Golgi apparatus given by Camillo Golgi, and ribosome George Palade, Fleming is by the Fleming given the chromatin structure. You can take a screenshot of this and also provide the notes. You can go through that. Here is the second question, guys. Mesosome in a cell is. Okay. Kindly go through the statements here given. One statement is correct. Okay. Membrane, option A, membrane bound vesicular structure. Option B, a chain of many ribosomes attached to a single mRNA. Option C, a special structure formed by extension of plasma membrane. Option D, a medium-sized chromosome. Yeah. Can you answer me, guys? You can answer me in the chat box also. And I will launch the poll. Yeah, I'm launching the poll. Yeah, which one is the correct answer? Very good, guys. Very good. I'm getting the correct answer. Very good. I think you have an idea about the mesosome. Very good. Very good. Very good, guys. Yeah, I'm sharing the result to you. Yeah, everyone answered C only, guys. Very good. Okay. See here. Membrane wound, I think you everyone answered correctly. No need of explanation. The chain of the many ribosomes attached to the single mRNA is called as polysomes. Right, guys? Many ribosomes are attached to the one mRNA. That is called as polysomes. Right. So that's why this option is wrong. Then coming to the medium-sized chromosome, no. And membrane bound vesicular structure, no. It is a special structure that is formed in the bacteria, right, of the plasma membrane. Can you anyone tell me what are the functions of mesosomes, guys? What is the main function of mesosome? Mesosome like function as a mitochondria in the bacteria, right? What are the functions of mesosome? Here you can go through this. This is the mesosome structure. You can see here the membrane bound from the plasma membrane extension it is. And here you can see the functions of the mesosome. It increases the surface area and it helps in the respiration. In the eukaryotes, mitochondria is there for the respiration, right? So here it is the having the respiration function, and it also helps in the replication of DNA and also reproduction, secretion of some of the enzymes required for the synthesis of the plasma membrane, and also the enzyme content, and also for the cell wall formation. Kindly remember this, all the things are the functions of mesosome. The main function is respiration. Here is the next question, guys. Here also match the following, and here you can see histones and nucleosomes and euchromatin, heterochromatin. You have to match with the list too. Okay. I've launched the poll. You can answer me in the chat box also. ATP production, not ATM production. Suddenly you got confused. Or typing mistake, I think, Sai Charan. Right? ATP production. Okay. I'm launching the poll. I think this answer you told actually. I'm launching the poll now. One second. Okay. You can answer me to this poll, guys. Yes, yes, yes. Everyone is giving correctly. D, D. Okay, Raju, very good. Easily you'll get the 4 marks, guys. Very easy questions, right? Okay, I'm ending the poll. I'm sharing the result here. You can see here. A is given by 20% and C by 10% and 70% is given D. So here you can see, guys, when we talk about the histones are the protein. Here you can see this is the DNA means DNA is negatively charged, right? Now, DNA has to be packed compactly means a positively charged protein has to be bind. Here you can see a histone octamer is going to be bind and the histones are positively charged. Can you tell me, guys, histone proteins are positively charged means which are the amino acid are there in the histones? Which amino acids are there in histones, guys? Giving the
Arginine and
positive, you can send me a message. Which all those? Lysine and arginine, right? Lysine and arginine are the very good. Arginine and the lysine. Very good, Raju. Yes, yes, Sai Charan. Lysine and arginine are the positive proteins. Lysine and arginine. Okay, are giving the positive nature for the histone protein. Then the for the A, it is B positively charged basic protein. Right. Then 1 minute. Yeah. And for the nucleosomes, you all know about the nucleosomes, right, guys? The DNA is wrapped around the histone octamer. How much base pair of the DNA is wrapped around histone octamer? 200 base pair. Remember, 200 base pair is wrapped around the histone octamer. And what are the histone protein, guys? Histone octamer. Which are the histone octamer? Histone octamers H2A, H2B, H3, and H4. This will form twice. So that is called as the histone octamer, which is wrapped around 200 base pair of the DNA. Okay, fine. Then coming to the euchromatin. Here you can see, 1 minute. Yeah, option D is the correct answer. Here you can see, guys. This is, you can see, the histone octamers were loosely attached. In the euchromatin, where it is loosely attached, whereas in the heterochromatin, it is densely attached. Okay. So that's why the heterochromatin is transcriptionally inactive. It will not do the any transcription. Transcriptionally inactive is the heterochromatin. Then coming to the euchromatin, where DNA is visible, right? Because loosely attached. So that's why it is transcriptionally active. Okay. Do you understood? Euchromatins are transcriptionally active, means transcription will taken place there. Heterochromatin, where nucleosomes are attached tightly, the histones are tightly attached. There is no transcription is going to be happen there. Okay. I think you understood these questions, right, guys? Okay. Can you tell me which histone is acting as a linker, guys? Which histone is acting as a linker? H1 histone. H1 histone is acting as a linker. Remember. Okay. Fine. Let us go to the next question. The DNA present in the chloroplast is whether it is a single stranded, linear, double stranded, circular, how it is. Very good, Sai Charan. How it is. I'm launching the poll. Yeah, you can answer me in the poll, guys. The DNA present in the chloroplast, you know, guys, chloroplast and the mitochondria is autonomous cell organelles, means they are having their own DNA and they are having an own ribosome, right? Then, which is the type of the DNA is present is, I'm going to stop the poll, sharing the result. Most of them given the 86% given the correct answer. Okay. The correct answer is circular double-stranded DNA. Right, guys. When we are talking about a chloroplast. Chloroplast is a double membrane, whereas the outer membrane is more permeable, guys. Okay. Whereas the inner membrane is a less permeable. Okay. Inner membrane of the chloroplast is less permeable. And inside that, it is having the a thylakoid membrane. A coin-like thylakoid membrane is attached. A bundle is called as a grana. A single coin-like structure is called as a thylakoid. And the cytoplasm of the chloroplast we call as a stroma. Okay. A dark reaction is going to be taken place in stroma. Light reaction is going to be taken place in thylakoid. And here you can see a chloroplast is having the DNA that is circular DNA and it is double-stranded. It is circular double-stranded. And it is having the ribosome. The ribosome type is 70S ribosome. 70S type of ribosome there. Okay. Fine. Let us go to the next question, guys. This is a statement-wise question. Given below, they are given two statements. Now you have to tell me whether which statement is correct or both are correct like that. Okay. Statement 1, mitochondria and chloroplast both double membrane bound organelle. Statement 2, inner membrane of the mitochondria is relatively less permeable as compared to the chloroplast. What they are telling is mitochondrial inner membrane is there, right? That is less permeable. Whereas chloroplast inner membrane is more permeable than that of the mitochondria. Which one is correct, guys? I'm launching the poll. No, guys, can you? Very good, very good, very good, Raju. Yes, someone told correct. Yes, Ruti Reddy, very good, correct. Raju can't give like this in the exams also, right? Be specific on one. Think once again. Read once again. Pa. Whenever you are getting confusion in this type of the statement-wise questions, you have to think once again. Okay. What are the things are going to be taken place in the mitochondria? In the mitochondria inner membrane, You've seen that ATP synthesis is going to be taken place. Yes or no? The ions are permeable easily, more permeable actually. The mitochondrial inner membrane is more permeable than that of the chloroplast. Now you can tell me which is correct. I'm giving a yeah. Yes, Sai Charan, very good. I'm ending the poll, guys. Sharing the results. See, most of them are given. A, both statements are correct only. Okay, fine. See here, guys. When we are talking about the mitochondrial chloroplast, the first statement is very easy. We know that statement 1 is correct. Okay. Then coming to the statement 2, here they are given. This is the mitochondria. Okay. See here. This is the mitochondria and this is the outer membrane and it is the inner membrane. This is the chloroplast. This is the outer membrane and it is the inner membrane. When compared to the chloroplast, the mitochondrial inner membrane is more permeable, okay, more permeable than that of the chloroplast. Okay, remember there is a so many things is happening inside the mitochondrial inner membrane. You know that in the inside of the mitochondrial membrane only where it is oxidation is going to so that oxidative phosphorylation is going to be taken place. Yes or no? Electron transport chain is there, right? So that's why inner membrane is more permeable in mitochondria than that of the chloroplast. So that's why here they are given the vice versa. So that's why it is wrong. So the correct answer is option B. So here you can see, sorry, option C is the correct answer. Okay. Option C is the correct answer, not option B. Then here is another questions, guys. How many different protein does the ribosome consisting of? This question is asked in 2023 actually. In the 2023 NEET exam, this question is asked. If you know, you can give me answer. If you don't know, means no problem. You know this, okay? Make a note of this. Very good. So many have given correct answer. Okay, fine, guys. I'm ending the poll, sharing the result. The correct answer is 83. Sorry, 83% given the correct answer. Yeah. The correct answer is D, guys. Yes, 80. Okay. 80 proteins are there in the ribosomes. Okay. You know that in the prokaryotes, we are having the 70S ribosome, and in the eukaryotes, we are having the 80S ribosomes. Okay. Fine. Here is the next question, guys. Which one of the following is not considered as a part of endomembrane system? Which one is not considered as the part of endomembrane system, guys? Okay, very good, very good. So many are given correct answer. I'm launching the poll. You can give me answer. Okay, most of them given the correct answer. I'm going to end the poll, guys. If you want to give the answer, give me fast. Okay. I'm going to end the poll, sharing the result. Yeah. So A is given by 13% and C is given by 88%. I think students are aware of this question. Fine. The endomembrane system, guys. The correct answer is A only. Okay. Not C, A. Okay. See here. Mainly, the double membrane organelle will not come under the endomembrane system. Okay. So that's why we are removing mitochondrial membrane and also the chloroplast and the chloroplast. Both are double membrane and peroxisome is also not comes under the endomembrane system. What is actually endomembrane system is the some organelles work together for having the same function, for the some function, productive functions. Here you can see the nucleus. After the nucleus, the rough endoplasmic reticulum will come. After the rough endoplasmic reticulum, the attached organelle you can see that is the Golgi apparatus. From the Golgi apparatus, the vesicles will be formed and the lysosome is also formed from the Golgi apparatus, right? So these all are working together. These all are working together. So that's why This part we are going to call as the endomembrane system. Nucleus, rough endoplasmic reticulum, Golgi apparatus, and the lysosome, and the vesicles also. Okay. Okay, then here is another question. Match the list 1 with the list 2. So here they are given the functions and you have to identify the organelles, guys. I'm launching the poll. You can go through this. I'm going to end the poll. Okay, fine. I'm sharing the result, guys. See here. Option A and B, 50-50%. You have confusion between option A and B. Okay, see here, guys. The correct answer is A. Okay. So when we talk about the nucleus, inside that there is a nucleolus is there. Okay. In the inside nucleus. Okay. This is the nucleus. Inside the nucleus, there is a nucleolus is there. Nucleolus is there. Okay. The nucleus inside the nucleus, actually, the ribosomal RNA is going to be synthesized. Okay. Inside the nucleus only, the DNA replication, RNA synthesis, all the thing is going to be happen. So here, the ribosomal RNA is going to be synthesized. Remember. Okay. Then coming to the centriole. Centriole, you can see here, mainly a cartwheels like structure. Organization, a cartwheels like structure is the centriole. Okay. Then coming to the Golgi apparatus, you know that the synthesis of the glycolipid, glycoprotein is the functions of, here you can see the function, the transport of the proteins and the secretion. And in the male sperm, you can see the acrosomes are present, right? Acrosome in the male sperm, right? So that is mainly synthesized by the Golgi apparatus, and plasma membrane modification is also done by the Golgi apparatus only, means synthesis of the glycoprotein and the glycolipid is the main function of the Golgi apparatus. Okay. Then coming to the leucoplast. In the plastate, there are three types will come. One is a chromoplast, means all the vegetables will look like a green color starting. Tomato will be green first, right? And after that, it will be looks like a red color. That is because mainly the chromoplast. Okay. After that, the chloroplast is converted into the chromoplast. So that's why it is going to be look like a red color. Okay. Then chloroplast, chlorophyll is there. So that's why it looks green color. Photosynthesis is going to be happen there. And the leucoplast. Leucoplast is mainly helpful for the storing of the nutrients. In the leucoplast, there are three types. It is very important. Amyloplast, it helps in the storage of the starch. Elioplast helps in the storage of lipid. And alluroplast, storage of the protein. Remember, guys, remember this. Alluroplast, starch storage. Elioplast, lipid storage. Alluroplast, protein storage. Okay. So that's why the correct answer is A. Okay, fine, guys. Let us start with the cell cycle and the cell division. Okay, what do you mean by cell cycle actually? We know that what the set of the all the processes which is going to be occur in the cell. For example, the cell is going to be formed, the cell is going to be mature, and the finally cell is going to be divide. Right. So this all the process together we are going to call as a cell cycle. Okay. So here you can see that this all the process together we call as a cell cycle. The time taken by the cell to divide, convert that one cell into that cell is going to be divide, right? How much of the time the one cell is going to be take to divide, that is called as generation time. For example, you are given the bacterial culture, you have taken only one bacteria. In within how many minutes the one bacteria is going to be divided? Okay, you know that this bacteria is going to be undergo the division, and this will give rise to the two bacteria by the mitosis. This is called as a generation time. How much time it is going to be taken place? 20 minutes. 20 minutes for bacteria generation time. Okay. And 90 minutes for the yeast, and 24 hours for the human cell. Okay. Human cell will divide. Human cell generation time is 24 hours. Okay. Whereas for the bacteria, it is 20 minutes, and yeast, it is 90 minutes. Okay. This is called as generation time. Then coming to the cell cycle, guys. Okay. Then how the cell is going to be divided? Mainly, the cell has to be divided means it has to undergo the preparation. Right. Now you all are giving the NEET exam, right? Simply, are you going to give an exam? No, right? You have to prepare yourself for the exam. You have to prepare for all the subjects, and you have to prepare 11th and 12th syllabus and all the thing, right? Like that, the cells has to prepare itself. So that's why there are two phases. First one is a preparatory phase. First you are going to prepare. Then second is you are going to give a exam, right? That is M phase. So totally two phase. One is the interphase and the M phase. In the preparatory phase, there are three phases, guys. One is a G1, yes, G2 phase. This G1, yes, G2 phase. This all the things will come under the interphase. Okay. Whenever the cell started the divide, 1 minute. Yeah. Whenever the cell started to divide, on the time you can see the cell is having the 2n number of the chromosome and content is 2C only. Okay. In the G1 phase, the content is same. In the S phase, it is going to be the DNA is going to be replicate. So that's why the content of the DNA is going to be changed. I am going to tell this in brief in the next slide. Okay, just see this slide. And in the G2 phase, it is going to be continued 2n and 4C. Okay, this is about the interphase. I am going to continue in the next slide also same. And coming to the M phase, in the the M phase may be mitosis or M phase may be meiosis. Okay, in the M phase, what is going to be taken place is karyokinesis and cytokinesis. Karyokinesis means the nucleus is there, right, inside the cell. The nucleus is going to be divided. In the cytokinesis, the cytoplasma and all the cell organelle is going to be divided between two. Okay, that is called as cytokinesis. Now let us see here. This all the cell cycle process is genetically controlled. Okay, it is genetically controlled. The protein name is cyclins. Okay, let us study in detail. So here you can see, guys, the G1 phase is the most longest phase. Okay, now you can see one cell is there. Now that one cell has to be undergo the division. Now what happened? The cell is having the DNA content is 2n and the DNA chromosome number, DNA content is 2C and chromosome number is diploid. Okay. Now what is happening here? In all the mitosis, it is diploid only. Okay. In the G1 phase, that is a growth first phase. Okay, in the growth first phase, the cell is going to be grow, right? So that's why in the G1 phase, the protein synthesis is more. Okay, the protein synthesis and RNA synthesis more here, and all the organelle is going to be duplicate because the organelle has to be shared between the two cells, right? So that's why all the organelles is going to be double here. Okay, then now what will happen? The cytoplasma is also going to be increased. Okay, the cell is increased in size now. Now coming to the S phase, in the S phase, the DNA is going to be double now. Okay, now the DNA is going to be double. So that's why in the S phase, the DNA content will shift from at the end of the S phase, not in the starting. At the end of the S phase, the DNA content will be 4C because the DNA is double now. But chromosome number is same only, 46 chromosome only. Okay, but the DNA content is increased here. So that is 4C. Okay. Now, after the S phase, it is entered into the G2 phase. Here, some of the autonomous organelle, that is mitochondria and chloroplast, is going to be double here. And the tubulin protein is going to be synthesis here. Okay. Why we require the tubulin protein, guys? For the mitosis, for the spindle fiber formation, we require the tubulin protein, right? For the next process, for the M phase. So that's why tubulin protein is synthesized in G2 phase only. Okay. And here you can see, guys, in the S phase, DNA replication, histone replication, and centriole duplication. These all are very important. It is going to be taken place in S phase. Okay. And after that G2 phase, then coming to the M phase. In the M phase, you know that M phase is it will take very less period of the time, only 1 hour in the humans. It will take only 1 hour. Remaining 90% is the interphase only, G1, S, and the G2. Okay. Then coming to the mitosis, you know that in the mitosis there are two phases. One is the karyokinesis and another one is the cytokinesis. In the karyokinesis, we can see these phases: prophase, metaphase, anaphase, and the telophase. Okay, fine. And you all know that some of the cells, are not going to be divided. For example, nerve cells, and also some of the heart cells is not going to be divided, right? So that's why they will go to the G0 phase. Okay, they will go to the G0 phase. Means once it is go to G0 phase, means it is not going to be divide again. It is not going to divide. It is not going to come to the cell cycle again. Fine. Then coming to the cell cycle checkpoints, guys. Cell cycle checkpoint means the cell cycle is controlled. Okay. So that's why. So here you can see three checkpoints are there. One is the G1 checkpoint. In the G1 checkpoint, It will check for the cell is having the good size for the continuing the cell division, and nutrients are there for going the replication, and growth factors all are there for the next processes, and whether the DNA is damaged or correct. If the DNA is damaged means it will not go to the S phase. Okay. It will go to the G0 phase. It will take some time and then it will enter to the G1 phase. Then it will continue the process. Okay. So for checking the DNA, there will be the checking. There is a checkpoint that is in the G1 phase. Okay. After the G1 phase, then the DNA replication is going to be happen and there is no checkpoint in the G2 phase. The G2 phase also the cell is going to be enter. When the cell is moving from G2 phase to M phase, there is a one checkpoint. Here, the checkpoint will check the size because the cell is going to be divided, right? So that's why the cell will check whether all the organelles are divided, whether all the organelles are there here, the cell is having the particular size and the DNA has been replicated or not. If the DNA is replicated, then only it will go to the M phase. Okay. Then it is entered to the M phase. Okay. In the M phase, it will check for the spindle assembly. In the M phase, you can see here, spindle fibers are correctly attached to the chromosomes or not. If the spindle fibers are not attached correctly means there will be a chances of aneuploidy. Aneuploidy. Okay. So that's why they will check for the whether the spindle fibers are attached correctly or not. Can you tell me guys, what do you mean by aneuploidy? What do you mean by aneuploidy? Kindly see, there will be a 3 checkpoint, G1, G2 and M checkpoint. There is no G2 checkpoint. Okay. And there is no S checkpoint. Sorry, there is no S checkpoint. Kindly see. Okay. Fine. Let us go to the mitosis, guys. You know that mitosis is a equational division, right? This is not a reductional division. The parental cell is also having the 2n and the daughter cell is also having the 2n. So that's why it is a equational division. Then coming to the, it is going to be taken place only in the somatic cell. All the parts of the cell, it is going to be taken place except the sex cells. Okay. Then coming to the mitosis is average of 30 minutes to 3 hours. And you know that the parental cells, the daughter cells are identical to the parental cell. And in the mitosis, there are two phases, karyokinesis and cytokinesis. In the karyokinesis, prophase, metaphase, anaphase, and telophase is there. Here you can see what are the things is going to be happen here. Okay. You all know that. Just imagine a cell is there. Okay. And in the cell is having the nucleus. First, we will only see the nucleus. Okay. So that's why what we will do is we will disappear all the organelles. Okay. Now, in the karyokinesis, all the organelles is going to be disappear now. Now, we concentrate on only the nucleus. In the nucleus, what will happen? The nuclear membrane is going to be disappear. Okay, and the nucleolus is going to be disappear. Nuclear membrane will become breakdown. Nucleus is going to be disappear. All the cell organelle has been disappeared. Now, the centriole is there, right? It is together now. It is moving towards the opposite pole. See here. Now, this all the both the centrioles is moving towards the opposite pole for making the spindle fiber. Okay, so that's why they are moving towards the opposite pole. Now, chromatin material become condensed. In all the interphase, the DNA is in the form of chromatin, right? Now, it will condense to form the chromosome, right? So that's why the chromatin material will condense now. Okay. Coming to the metaphase, the spindle fiber will attach to the kinetochore. See here, in the chromosome, here the centrosome, there is a kinetochore will be there. For the kinetochore, both the spindle fibers are attaching and chromosomes will to move toward the center place. That is called as equatorial plane, where the chromosomes move toward the equatorial plane for forming the metaphase plate. Okay. Now, in the metaphase plate is going to be formed. Okay. And here you can see that the metaphase is the best stage to study the chromosome. Okay, because it is condensed correctly now. That's why. Then coming to the anaphase. In the anaphase, the centromere is going to be split. See here, guys. Now the chromosomes are attached in the attached now, right? Like this. These are the two poles. This side is one pole. This side is one pole. Now these are attached, right? Now the chromatids is going to be split. Now chromatids is going to be split here, and due to the pressure, this chromatin will come this pole and this will move to the opposite pole. So this will look like the anaphase. The chromatids are in the opposite phase, okay, opposite pole. So that is called as the anaphase. Now the centromere will split up and it is going to be moved towards the opposite pole. Okay. Okay. And here is the best stage to study the shapes of the chromosome, because the chromosomes will look like in the different shapes V, L, J, and I. Okay. Then coming to the telophase, guys. In the telophase, what are the things happened in the prophase will be repeated again, means opposite to that. Okay. Opposite to that. What is happened there? Nuclear membrane is disappeared, right? But here nuclear membrane is forming. And nucleolus will disappear there, it is forming again here. And all the cell organelle disappeared there, here it is appear again. And chromatin there, it is become condensed. Now here chromosome is there, it is become decondensed to form chromatin. Okay. Now you can see two cells are there. Okay. This is about the mitosis. Do you understood this? Okay. Here you can see, guys, you can go to this cytokinesis uh, slide. I will give you the notes to you. In the cytokinesis, there will be a two types, guys. There will be a cell furrow method. This is also called a centripetal. That will be take place in the animals. Animals. Okay. Centripetal will take place in animals, whereas the cell plate formation, that is like the centrifugal. Centrifugal. This will take place in the plants. Plant. Okay. See here. In the animals, the cleavage will start from the peripheral region, then it will goes to the center, and all the both the cells is going to be separate here. You can see here like this. Okay. Whereas in the plants, it is different. The some of the vesicles formed from the Golgi apparatus and the ER vesicles that will be deposited in the center, and it will move toward the peripheral region. It will deposit in the center, then it will move towards the each pole, then each side, then the cell is going to be separate. Here you can see the cell is going to be separate. This is called as a centrifugal method or the cell plate method in the plants. Okay. Fine. We'll go to the meiosis, guys. You can see meiosis we have to explain here, but we are running out of the time. Once again, I will tell this in the tomorrow's class. Okay, when we are talking, I will launch the poll, guys. You can give me your feedback. Yeah, I'm launching the poll. You can give me your feedback, guys. You can go through this PPT I will give you. Okay. And here there are some questions I have attached here. Kindly go through this all the questions. In the meiosis, mainly the first stage of the meiosis, that is the prophase, that is consisting of the leptotin, zygotin, pachytin, and the diakinesis. Those all are very important. You can go through that. Okay. Very good, Shruti. Yes, we will get the chromosomal disorder if the aneuploid is happened means. This concept is very easy, guys. Okay, this concept is very easy. You can go to this chapter. Okay, thank you so much. Can give me your feedback, then you can log out.